Hello students, I am Dr. JAYASHREE, Assistant Professor, Department of Biotechnology, School of Bio and Chemical Engineering, Satyabama Institute of Science and Technology. Today our topic is CAMP and phosphatidylinositol signaling pathway. These pathways are important in G protein coupled receptor. So coming to the introduction about CAMP. What is mean by CAMP? CAMP is cyclic adenosine monophosphate, is an important secondary messenger in many of the biological process and are associated with many cell signaling transduction pathways. These include increase in the heart rate, cortisol secretion, it's a type of hormone, it's reduce the stress level, and then breakdown of glycogen and fat. Subtypes of G alpha and G alpha subunits like GS and GI either activate or inactivate the adenylyl cyclase enzyme which convert adenosine triphosphate. In previous video we have discussed about G protein coupled receptor. In that we have talked about alpha subunit GI and GS. This will be whether they will be activate or inactivate the adenylyl cyclase, so which will help to convert the ATP to cyclic adenosine monophosphate. So if the it helps, what is the role of CAMP? So it helps to stimulate the CAMP dependent protein kinase A. This protein kinase A is a tetramer enzyme, consists of two regulatory subunit and two catalytic subunit. To activate the enzyme, two molecules of CAMP will go and bind with the regulatory subunit and trigger the conformational stage and dissociate the complex. So, the CAMP binding to the regulatory subunit of protein kinase A and allowing to release the catalytic subunit of protein kinase A. What is the role of catalytic subunit? These catalytic subunit of PKA bind to the SERB. SERB is nothing but CAMP responsible element binding protein. These proteins can be phosphorylation of the CAMP responsible element protein. It regulates the diverse cellular response like proliferation, survival and differentiation. This image can clearly explain you the CAMP pathway. First step, the GPCR undergo conformational change upon ligand binding. The ligand is binding to the GPCR. It will activate the G protein. This is the inactive form of G protein. When it is activated, the GDP will be converted to GTP. I already mentioned this everything in the G protein coupled receptor with this in a previous video. Alpha bounded with GTP will go and bind with adenylyl cyclase enzyme. This adenylyl cyclase enzyme will convert the ATP to cyclic AMP. This cyclic AMP directly go and bind with the protein kinase A. Protein kinase A is a tetrameric enzyme. It contains two regulatory subunit and two catalytic subunit. So when the CAMP is going and two CAMP molecule will go and bind with regulatory subunit, so the PKA will be coming to the active form. When it is active form, the regulatory subunit and catalytic subunit will be separated. This catalytic subunit will go to the nucleus and bind with the SERB protein. SERB is nothing but CAMP responsible element binding protein which will be cursing gene expression that mediate the cell growth and differentiation. These are all the important steps in CAMP pathway. Phosphatidylinositol signaling pathway, it regulates the various cellular process such as protein proliferation, growth, apoptosis and cytoskeleton rearrangement. Here again the alpha subunits have GQ and GO will be playing an important role. They will be alternatively activate the phospholipase C enzyme. This enzyme helps to hydrolyze phosphatidylinositol 4,5-bisphosphate PIP2 into diglycerol TAG and inositol 1,4,5 triphosphate IP3. So it will be this enzyme help to hydrolyze the PIP2 into DAG and IP3. This, what is the role of? First we will see the IP3 role. IP3 will be going to binding with the endoplasmic reticulum. There will be an ion channel present in the endoplasmic reticulum. So it will trigger the ion channel to release the calcium ions into cytosol. This calcium control the activity of numerous downstream target proteins present in the cell. One of the important downstream target is protein kinase C. Calcium allow protein kinase C to bind with DAG. DAG is activate the protein kinase C activity. It helps activate the many target proteins. Then PKC, then phosphorylate downstream substrates such as glycogen synthase and calmodulin protein. All these are the important steps in phosphatidylinositol signaling pathway. This image can explain you about the phosphatidylinositol signaling pathway more clearly. First, the phospholipase C enzyme will be activated by alpha subunit alpha Q. It will activate the phospholipase C. It will go and bind with PIB or activate the PIB2. It will be separated into DAG and IP3. This IP3 go and bind with endoplasmic reticulum, and it will endoplasmic reticulum calcium ion channels and open the channel, and it will release the calcium ions to cytosolic place. These calcium ions then go and bind with the protein kinase C, which will be present in the cytoplasm. So, this protein kinase C will go and bind with the DAG, which will be present in the plasma membrane, and activate the DAG. So, this PKC and DAG will phosphorylation of substrate. So, it will be helpful for the many of the downstream process which will be happening inside the cellular process. So, then these calcium ions also able to bind with ion channel which will be present on the membrane, plasma membrane, and will help to send the calcium ion out of the extracellular space. This is the full explanation about CAMP and phosphatidylinositol signaling pathway. I hope this video will be helpful for you. These are all the reference images and papers I have taken. Thank you all.